All right, we are live with our final session of today's day 2 of EscarpCon. We're talking about controlling flavor, and for our final topic, we're going to be talking about pretty much one of the hardest things that we can control in beer, and that's Brettanomyces. One of our favorite yeasts at Escarpment Labs, and part of the reason that we love it so much is that it can do so many different things, but also part of the reason that we hate it so much is that it does all these different things that we can't predict, and that we've really struggled to control. So when we started to be able to collaborate with the University of Guelph here in Guelph, the lab of George van der Merwe, it was really exciting because we could start to study these things. And one of the things that we found is we had all these Brett strains, but we knew nothing about them. We knew nothing about, what are the right conditions to grow them? What flavors do they make? How do we combine them with other yeasts to get ideal flavor profiles out? And that's where Caroline here came in. Caroline came into that lab as a undergrad student and expressed some interest in all this crazy beer stuff that we were talking about, and ended up sort of sticking around and doing a master's project on Brettanomyces in brewing. And I think Caroline was able to make a very meaningful contribution to sort of this side of beer research, and really helped to push things forward. So I was really excited to ask her to come on and talk about some of that work that she did over at University of Guelph. Caroline is still a researcher at the University of Guelph, helping out keep the show on the road for that lab. They sort of spend half their time studying yeast metabolism in its fundamentals, and about half their time studying beer and wine and cider and all the fun tasty things we can do, and how we can optimize yeasts for producing those beverages. So really fun to collaborate with these people. I'm really glad that Caroline was able to stick around and continue to contribute to research. And she's been doing some stuff with Kvike and cider. And actually, maybe you can fill us in on what you're working on these days, Caroline.
Yeah, so most of the time these days working on the beer fermentation side in our lab. A lot of times doing Kvike research more so. So looking at sort of their metabolism, how they behave, all of their stress adaptations, things like that. Also helping out a bit with the cider projects in the lab, sort of anything on the industrial side.
Awesome. For those that are joining us live, please feel free to start your discussion on Crowdcast. Feel free to ask questions. We love getting these questions and trying our best to answer them at the end of these webinars. And then for those who are tuning in after the live cast, thanks for checking out what we have to offer. Hopefully we'll see some of you live in here one day. And just wanted to mention that a recorded version of this webinar will be available on Crowdcast and YouTube. So if you're busy doing stuff right now, don't worry. You can check this out afterward as well. But without further ado, I want to give Caroline the opportunity to tell us about demystifying Brett.
All right, thanks for the introduction there, Richard. Let me just share my screen here. All right. So, hi, everyone. As Richard mentioned, I'm a researcher at the University of Guelph, and a couple of years ago, I finished my master's thesis on Brettanomyces bruxellensis. It was quite a fun 2 years. I learned a lot, and I'm excited to kind of take you guys through what I managed to find out about this yeast. So before we sort of delve into all of my research and everything I learned, I just wanted to give a really quick overview in case anyone hasn't worked with this yeast before. So Brett is a yeast that has gained importance in the craft brewing industry recently. So beer fermentation is most often associated with sack or brewer's yeast, but consumers are always kind of looking for something novel, something unique, and brewers are trying to find a way to bring that to them, sort of stand out from the crowd. And a way to do that is with non-conventional yeasts that are capable of fermenting. Brett is an example of this sort of yeast. And out of all of these non-conventional yeasts, Brett has become the most popular in recent years. So it actually shares a similar niche to that of Saccharomyces cerevisiae, but the two yeasts diverged from each other approximately 200 million years ago, and they actually evolved to have these fermentative capabilities independently of each other, which is really interesting. Amongst its closest relatives, Brett is actually the only one capable of fermentation. And it also has a few unique properties that make it sort of different and interesting to work with compared to sac. One of those is that it's capable of metabolizing a wider variety of sugars. So you can actually produce what's called a super attenuated beer, one with a slightly higher alcohol content and also a slightly drier product, less residual sugars left over. And in addition to that, it produces flavor metabolites that SAC is not capable of producing. So you can create sort of unique novel flavor profiles with this yeast. So I just want, on the left-hand side here, I just have two images just to sort of give you an idea of how different these yeasts can be from each other, even just from sort of a morphology perspective. At the top here, there's Saccharomyces cerevisiae, and these are what these cells look like under a microscope. And at the bottom is the Brett cells. And you can kind of see right away by looking at these that the SAC cells are quite a bit larger than the Brett cells and sort of more round in shape. The Brett cells are more lemon-shaped. And that just has to do with how these yeasts bud off from each other or separate from each other as they grow. So up until recently, Brett got a pretty bad rep as a wine spoilage organism. This is what it was sort of most well known for amongst people. On the right-hand side here is a flavor wheel with sort of flavors that were attributed to Brett in a wine environment. And just to give you a bit of an idea of some of the flavors that are on here, there's things like Band-Aid, wet dog, urine, leather. Not exactly something I'm looking for in my wine, probably not something you're looking for either. So for a really long time, the majority of the research that was done on this yeast was just trying to minimize those off flavors and trying to make sure that we minimize the growth of Brett in wine. This came with a pretty big consequence though, and that very little research was being done on the biology of this yeast, how it fermented, what sort of flavors it was capable of producing. And so the research kind of stalled out on that side for a long time. And then recently it became popular in the craft brewing world. People became interested in it again. And as a result, research is sort of shifting now. We're starting to look at those things we didn't know about it in a more positive aspect, which is really awesome and really cool to start seeing. And the whole reason for that is that Brett finally found its calling in the craft brewing world. You know, the flavors that are associated with it are considered positives in many styles of beer, particularly the Belgian styles such as lambics and saisons, but craft brewers are also starting to look at it in different styles of beer as well. And the reason for that is it can produce those compounds that Saccharomyces can't. It's been used in a number of craft breweries. At the bottom here, I have just 3 examples from local Toronto breweries. There's Brett Peach from Burdock, Barn Owl from Bellwoods, and Flemish Cap from Folly. These are just some examples of how it's being utilized in this world now. So Brett is, like I've been saying, you know, has those flavors that are very unique that SAC can't produce. They were oftentimes described as funky and fruity, and together they're referred to as Brett character. So the funky flavors are due to compounds called phenols, such as 4-ethyl glycol and 4-ethyl phenol. 4-ethyl glycol, or 4-EG, gives off sort of a smoky clovey aroma, and 4-EP gives off a barnyard aroma, which sounds very weird, but it works quite well in a lot of these beer styles. And the fruity flavors are due to compounds called esters. And those are compounds such as ethyl caprylate and ethyl caproate, which give off a pineapple or a pineapple red apple sort of aroma. And these together, you know, allow for novel kind of unique products to be made that can be different than those that can be produced with Saccharomyces strains. But anyone who's ever worked with this yeast knows that there's also a number of frustrations and challenges that come along with working with it. It's an extremely slow grower, which was the bane of my existence during my master's. It took forever to get anything done just because just growing it up and fermenting with it took so much time. And this led to unpredictable fermentations. You never knew how long things were going to take. And this leads to a real problem for brewers as well. You're tying up fermenter tanks. You can't sort of ferment things as quickly or get products out quite as quickly. It leads to higher operating costs. And it's one of the things that sort of was this mystery with Brett. So the reason for this sort of slow growth is that when we see Brett grown in an aerobic environment, such as a pre-growth generally is, it performs quite well. It can, you know, start metabolizing sugars. It starts producing biomass, ethanol, and some acetic acid. But then once you transfer it to an anaerobic environment, like that in a fermentation, you see it kind of stop. It has this lag phase where it's not consuming sugars, it's not producing any sort of metabolites or anything like that. And it takes several hours or sometimes even days for it to get out of this phase and to continue the fermentation. And this was the reason that, we have these lengthy fermentations, and one of the main things we're trying to find an answer to. So over the years, in past research, there was a number of things that were tried in order to alleviate this lag phase, but I think just by looking at this list, you'll see why many of them sort of aren't an option for the brewing world. There's things like acetaldehyde, formaldehyde, acetone, acetoin, which can be carcinogenic, toxic, or have sort of these aromas that you're not really looking for in a beer, you wouldn't want to have. And then as well, there's oxygen, which does work quite well, but it eliminates that anaerobic environment that you need in a fermentation. And it also causes an increase in acetic acid production. So none of these are really viable options that brewers can use to speed up their fermentations. So based on all of this background research, we were left with two questions when I started my master's thesis. The first one was, can we better understand the fermentation kinetics and flavor compound production exhibited by this yeast? And the second one was, can we alleviate the lag phase experienced by this yeast through supplementation of nutrients that will not detract from the flavor profile and will also produce a product that is safe to consume? Which brings me to the research that was done during my master's thesis on this yeast. So first, before I get into the sets of experiments I did, I just wanted to take you through our fermentation workflow that we use. We work in volumes that are quite a bit smaller than the industrial scale would use, but they work quite well for us. We're limited with the space that we have. So the way that we start this is, first off, we streak out our strains onto WLN plates, grow them up to isolated colonies. From there, we pick an isolated colony and we inoculate that into YPD broth, which is then grown at 30 degrees Celsius with shaking. Once that is finished growing, we transfer it to a wort pre-growth, which is also grown at 30 degrees Celsius with shaking to get our yeast acclimatized to those wort sugars before we also throw them into an anaerobic environment, which provides an additional stress. Once these yeasts finish growing, they're then transferred into the wort fermentations. And the way we do these in our lab is in spice jars with 70 milliliters of wort, and they're fitted with an airlock on top. So these fermentations are weighed throughout the process, these spice jars, to kind of track the rate of fermentation, how well the fermentation is progressing. So as the yeast ferment, they produce carbon dioxide, which is then released through the airlock, leading to a weight loss in the fermentation. So the more weight loss that is occurring, the more carbon dioxide that was produced, and the higher the rate of fermentation is. Following the fermentation, we do post-fermentation analysis, such as GCMS to measure the flavor compounds that are produced, and HPLC to measure the sugars content and the ethanol content of the final product. So the first experiment that I did during my thesis was primary, secondary, and co-pitch fermentations to try and see how Brett performed in combination with Saccharomyces, how it affected the flavor profiles developed by Saccharomyces. And these were done at 22 degrees Celsius in a standard ale wort, and they were carried out for 21 days. So for the primary fermentations, we wanted those as sort of a baseline so that we knew how each Saccharomyces and how each Brettanomyces performed by themselves in a fermentation, what sort of flavor profiles they developed, how quickly they could ferment, so that when we then moved on to secondary and co-pitch fermentations, we would know sort of what effect Brett had on the Sac strains, because we had a baseline. So for the primary, we inoculate Saccharomyces or Brettanomyces at the start of that fermentation and let that progress throughout. In a secondary fermentation, we inoculate Saccharomyces at the start, let it ferment for 7 days, and then inoculate Brettanomyces at a lower pitch rate. And then for the co-pitch, we inoculate both the Saccharomyces and the Brettanomyces at the start of the fermentation, but at differing pitch rates. So these are the strains that we used, 3 Brettanomyces strains and 3 Saccharomyces strains. There's experiments done prior to this that allowed us to sort of choose which Brettanomyces strains we wanted to work with from a greater list that we had. And this was based on experiments we did, and we sort of picked the best strains for the 2 categories that I'll talk about. So we chose BSI Dre as sort of our standard Brett strain. It's a very commonly used Brett strain in the industry. So we wanted that as sort of our baseline. And then we chose BBY11 as our beer isolate, had a good fermentation profile, and it also had a good flavor profile. And then we chose Pest 4 as our wine isolated Brett strain. We wanted to be able to compare how a beer isolate and a wine isolate performed in comparison to each other, and if there was any difference in how they fermented and what sort of flavors they produced. And then we also had 3 SAC strains. We had Cali, which has sort of a neutral flavor profile and is POF negative, Vermont, which has a fruity profile and is also POF negative, and St. Remy, which has a more phenolic flavor profile and is considered POF positive. And we just wanted sort of a range of different flavor profiles for our Saccharomyces strains. Then following that, we have the pitch day and rate. So like I said, for the primary fermentations, both Brett and Sac were pitched right at the start with a pitch rate of 12 million cells per milliliter. And then Sac was pitched into secondary and co-pitch also at the start at 12 million cells per milliliter. Brett in the secondary was pitched at day 7 with a lower pitch rate of 1 million cells per milliliter, and in co-pitch, it was pitched at the start as well, but also at that lower pitch rate of 1 million cells per milliliter. So then moving on to the results, these are the fermentation profiles for our primary fermentations. And again, these were just to create a baseline of how each of these yeasts performed. And we got fermentation profiles that are very characteristic of both Brett and Sac. This is what we were expecting to see. Right at the start here, these 3 more green yellow lines are the Saccharomyces fermentations, and we see those fermentations take off right at the start here, ferment quite well, and sort of begin to plateau out around day 5 or 6. The Brettanomyces strains, you see a lag phase at the start here where it's not really doing much for the first day, day and a half before the fermentation takes off, and then has a slower progression. You never quite see that same rate of fermentation at the start that you do with Sac. However, it progresses quite steadily, and by the end of the 21-day fermentation, we see similar degrees of CO2 evolution or weight loss in all of the strains. Following that, we did a secondary fermentation, and here we see that at the start, we have Saccharomyces pitched in. It is able to do its fermentation, completes it around day 5 or 6, and then we have Brett added in at day 7 right here. And after a short lag phase, we see it take off, and it is able to complete a secondary fermentation following the initial Saccharomyces fermentation. What we see here is quite a difference in how these strains ferment. We see that strains that had Cali as the primary strain performed worse than some of the others in a sense where they produce less CO2 by the end of the fermentation, whereas those with St. Remy as the primary strain, these 3 at the top, perform the best with the highest CO2 evolution seen here. For co-pitch fermentations, we had both Brett and Sac pitched at the start there, but at different pitch rates. And we don't really see much of a difference in terms of these fermentation profiles. We see all the fermentations take off at the start here and sort of finish out around, day 6 or 7 with some slight increases here and there. And we don't see a wide range of CO2 evolution depending on the strain combination. All of them seem to perform fairly well. And then for the ethanol concentrations, these were measured with HPLC at the end of the 21-day fermentation. And what we see here is that the ethanol concentrations in secondary and co-pitch fermentations are higher than that in the primary fermentations, which is what we would expect. Like I mentioned at the start, Brett is capable of metabolizing a greater number of sugars compared to Sac, which is why we likely see this slightly higher ethanol concentration. The reason we might not be seeing that in the primary fermentation here is that 21 days is a bit of a short fermentation for Brett, so it might actually not have finished its fermentation yet and might need a few extra days to fully metabolize all those sugars. And then I pulled out just a couple of compounds out of our list of flavor compounds, just to give you an idea of some of the trends that we saw. So at the top here, we have vinyl guaiacol, and then at the bottom, ethyl guaiacol. And what sort of stands out right away is that in both secondary and co-pitch, the levels of the 2 compounds are lower than that in the primary fermentation, which makes you think that the secondary and the co-pitch fermentations are less funky than that of the primary. We also see something that's very characteristic here where The Saccharomyces strains, which are the 3 at the start here, produce vinyl glycol, but they're not able to convert it to ethyl glycol like Brett is capable of, and so we don't see levels of that here. And for the ester compound profiles, we see a similar pattern to that with the phenol compounds, where both the secondary and the co-pitch fermentations have lower levels of both of these compounds in comparison to the primary fermentations. We see much lower levels, especially in something like ethyl caproate, where the levels are actually falling quite a bit lower and below threshold in some cases in comparison to the primary fermentations. We also see that the Brettanomyces primary fermentations are quite a bit fruitier than that of the Saccharomyces primary fermentations for both of those compounds. And then this is a heat map. It's a lot of information on one slide. This is for all of our combinations and all of our flavor compounds for this experiment. But the thing I want to draw your attention to is these 2 bars on the left-hand side here. The one on the left is the fermentation style, and the one on the right represents the primary strain. And the way this diagram works is that it clusters based on the fermentation style and the primary strain, those that are most similar. And what we see here is that despite the influence of Brettanomyces on the flavor profile developed by the Saccharomyces strain, primary strain, actually still plays a big role in the final flavor profile, as we can see groupings based on the primary strain here. So for example, St. Remy co-pitches, Cali co-pitches, then there's Vermont secondaries at the bottom here, all clustering together. The other thing that I need to say for this is that there is a possibility that because it was only a 21-day fermentation, Brettanomyces flavor profile might not have been fully developed, which could be why the primary strain is playing a larger part in the final flavor profile. However, it's really hard to sort of guess at that. So the next experiment I did for my thesis was a time course experiment. And for this, I was curious to see at what point during a fermentation different flavor metabolites were being produced, and then at what point all the different sugars were being consumed and ethanol was being produced. So the way I did this was I set up the spice jars again, but had different ones for every time point. These fermentations were again done at 22 degrees Celsius for 21 days. And I had time points at day 0, and then days 1 through 5, days 7, 10, 14, 17, and 21. And I took time points at each of those for both HPLC and GC. So in this graph here are the results from HPLC, and we see all of the strains represented here. I took a subset, so I did Cali, BSI Dre, and BBY11 primary fermentations, which are these 3 at the start here. And then I did a co-pitch fermentation of Cali and BSI Dre and Cali and BBY11. So I wanted to keep it simple. There's a lot of jars involved in this experiment, so I chose just one Saccharomyces strain, kind of the standard one we use in our lab, which is Cali, and then just the 2 beer isolated strains, which seem to perform better than the wine isolated Pest 4 strain. And we wanted to do it in co-pitch, as that allowed for both strains to be added in at the start, which just made it a little bit easier. And then what we see here at the top is glucose. And what we see is that for all of the strains and all of the combinations, glucose is fully consumed by the end of the fermentation. And actually for any of the fermentations that had Cali in them, so the Cali and the 2 co-pitches, it was fully consumed by the end of the first day of fermentation. It was slower for BSI Dre and BBY11, which again we would expect since they are slower growers and slower fermenters. And we see more of a lag phase at the start where the concentration between day 0 and day 1 doesn't really change for BSI, and it doesn't really change for the first 2 days for BBY11. Following glucose consumption, maltose was the next sugar to be fully consumed by all of the fermentations apart from the BSI Dre primary fermentation, which we see here. We see that all of it's consumed quite quickly in all of the others within the first 2 weeks of fermentation. And then the last sugar is maltodextrose, which the strains seem to struggle with a bit more. Only BBY11 over here and the Cali BBY11 co-pitch was actually capable of consuming all of the maltodextrose by the end of the fermentation, we still see levels of it in the other fermentations by day 21, particularly in BSI Dre, where it's quite a bit higher. And then in terms of ethanol production at the bottom here, we see quite a bit of variance in how much ethanol was produced for the different strains. Please note that the y-axis is different for the graphs, which makes it a little confusing, but it is leads to quite different levels of ethanol in these. And something that was quite interesting was that despite the fact that BSI Dre sort of struggled with the sugar utilization more than BBY11 did, they both produced similar levels of ethanol. So, which leads us to believe that BBY11 might be using that sugar for something other than producing ethanol. Could be something like biomass production, flavor compound production, or as a storage metabolite. And then we also see that for Brettanomyces fermentations, the level of ethanol generally peak near the end of fermentation for both of these, whereas for the Saccharomyces fermentations, the ones containing Cali, it peaked closer to the start of the fermentation here. And then for the flavor production timeline, I picked out just one ester and one phenol out of our group to keep it simple and sort of illustrate the trends that we saw with these flavor compounds. So on the left-hand side here is ethyl caprate, which is an ester, that fruity sort of profile. And what we see here is that production is higher in the Brett primaries in comparison to the Saccharomyces primary and the co-pitches, which is also what we saw in that previous experiment. So this just confirmed those results again. And we see the highest production or the highest level, sorry, at day 14 for all the strains. And then those levels diminish a bit as we move further into the fermentation. And the levels are quite a bit lower for these strains here. And then for ethyl phenol on the right-hand side here, which is the phenolic funky compound, we see production of this compound starting right at the start of fermentation, right at day 1, for all of the fermentations that have Brett in them. Again, we see that levels of this compound are generally higher for the primary fermentation in comparison to the co-pitch fermentations, and we don't see production really in the Saccharomyces strain, which is to be expected. And again, the levels sort of vary throughout, and despite the fact that they start being produced at the start, the highest levels tend to be near the end of fermentation, which has been shown in literature before. These compounds tend to peak in terms of concentrations when these cells are further along in their growth. And then the last set of experiments that I did for my thesis was supplementation fermentations. And this is where I tried supplementing with various nutrients in an attempt to see if we could alleviate that lag phase or have a quicker or a better fermentation rate for various fermentations, while also adding in nutrients that wouldn't sort of become an issue for brewers. So not something that would be considered toxic or affect the flavor profiles, hopefully. So I'll just take you through the supplementation fermentation workflow. It's a little bit different than the one for the other fermentations. So first off, again, a single colony was inoculated into YPD just as before. But once that was finished growing, it was then split up into a wort pre-growth and a wort pre-growth containing this nutrient supplementation. Following growth of that, each of the pre-growths was further split up into a wort fermentation and a wort fermentation containing the nutrient supplementation. And this left us with, This left us with 4 combinations. So we had no supplementation in pre-growth and no supplementation in fermentation as our control. And then no supplementation in pre-growth, but supplementation in the fermentation, supplementation in the pre-growth, but no supplementation in the fermentation, and supplementation in both pre-growth and the fermentation. And these were again done at 22 degrees Celsius, but for 28 days because I only did these as primary Brettanomyces fermentations with our 3 strains, BSI Dre, BBY11, and Pest 4 that I used for prior experiments. And these are the supplementations that I did for my experiments. I did thiamine for a final concentration of 0.48 milligrams per liter, as well as 3 individual amino acids, aspartate, glutamate, and glutamine, each at a concentration of 1.05 milligrams per milliliter. So these are the results from the thiamine fermentations. You see each of the 3 strains here. So just focusing on the BSI Dre on the left-hand side here, we see that supplementation in both the pre-growth and the fermentation, this blue line, results in the highest CO2 evolution by the end, so the best fermentation rate. We don't really see an alleviation of the lag phase for any of them here. However, it is interesting that the supplementation did allow them to sort of ferment better. For the BBY11, we see the highest CO2 evolution for supplementation in the pre-growth, but not the fermentation. And we also see that the other supplementations, whether that be in both pre-growth and fermentation or just fermentation, led to higher CO2 evolution in comparison to the control, which is the yellow line there. We don't see any sort of benefit in terms of alleviation of the lag phase at the start here. They all seem to start fermenting at the same rate. And then for PES4, we also see the highest CO2 evolution for supplementation in pre-growth, but not fermentation. And we see a slight alleviation of the lag phase here for that strain, where we see sort of a quicker takeoff to fermentation in comparison to the other combinations. Something that's interesting here is that when you look at all 3 of these strains, we don't really have the same order of sort of least effective to most effective treatment. All of them have a different order here. Some of them prefer, supplementation at both stages. Some of them prefer supplementation at just one stage. So there isn't really a one-size-fits-all here. It's sort of strain dependent, and we need to take that into account. Moving on to the amino acid fermentations, these are the results for BSI Dre. So on the left-hand side here is supplementation with aspartate. And what we see here is that supplementation at any stage, whether that be in pre-growth, fermentation, or both, resulted in a higher CO2 evolution in comparison to the control, which is this yellow line here. And we also see that supplementation in both pre-growth and fermentation, which is this green line, and supplementation in just pre-growth, which is a sort of blue aqua line, resulted in a bit of an alleviation of lag phase, where we see the fermentation take off quicker for those 2 combinations in comparison to the others. In terms of glutamate, we see the highest CO2 evolution for supplementation in both pre-growth and fermentation. However, it's pretty negligible in comparison to the control, the yellow line, which it's pretty much right underneath there. The significance is probably negligible. And we don't really see an alleviation of a lag phase at the start here for any of the supplemented fermentations. And for glutamine, we see the highest CO2 evolution for this fermentation that had supplementation in the pre-growth over here in comparison to the others. We do see a slight alleviation of lag phase for the fermentation that had supplementation added to it, which is this blue line here in comparison to some of the others. But based on these results, it seems that aspartate had the greatest effect on the fermentation for BSI Dre, regardless of when the supplementation was added in or whether it's at both stages, as all of those fermentations performed better than the control. For BBY11, we again see that supplementation of aspartate at any stage led to greater CO2 evolution than the control by quite a large margin here, but no alleviation of the lag phase at the start. For glutamate addition, we also see that addition of any of supplementation at any stage results in higher CO2 evolution in comparison to the control, as well as with glutamine, we also see that result. And with glutamine, we see an alleviation of the lag phase occurring quicker with supplementation and fermentation compared to the others. So overall, it looks like supplementation of any of these amino acids would lead to a benefit with BBY11, as they all performed better than the control here. And finally, for pest 4, we see pest 4 performed differently or had different results than we saw for BSI Dre and BBY11, which was pretty interesting. We don't really see as much of a benefit from addition of any of these amino acids as for all 3, aspartate, glutamate, and glutamine. The control actually had the highest CO2 evolution for all of them, this yellow line here. We do see a couple of other interesting things, though, that could be of note. Supplementation in pre-growth of aspartate led to an alleviation of the lag phase. However, it did not produce higher CO2 evolution. But that is still an option for alleviating the lag phase at the start there. And then as well for glutamine, we see an alleviation of the lag phase with supplementation in both pre-growth and fermentation. And that could be beneficial as that was also the fermentation that had the second highest CO2 evolution, and it's not too far off from that of the control strain. So it's interesting to note those differences and how differently the wine-isolated Brett strain performed in comparison to the beer-isolated Brett strains. So this is sort of just a research summary of the main key findings from my master's thesis. We see that niche adaptation plays a role in fermentation efficiency and flavor compound production. We see differences in comparison of the beer isolated strains of Brett compared to those of the wine isolated strain. And we see differences as well in terms of how nutrient supplementation affects strains depending on where they were isolated from. We also see that secondary and co-pitch fermentations are effective in developing Brett character on a shorter time frame. So if you want to have some of those flavors that are associated with Brett, like those phenolic compounds or those fruity compounds, but you don't want to have a month plus fermentation, then doing a secondary co-pitch fermentation will still develop some of those flavors and you can have a shorter fermentation. As well, benefits of nutrient additions are strain dependent. There isn't sort of a one size fits all for them or a nutrient cocktail we can create and say this is how it's going to work for every strain. It's definitely going to have this benefit. Which leads me to the major key finding of my thesis is that you really need to know your strains. No two strains are alike, despite the fact that they both come from a wine background or they both come from a beer background, or they're both Brett strains or both SAC strains. They're going to perform differently. They're going to have different nutrient requirements, and we really need to treat them independently and get to know your yeast before you sort of expect it to do something for you. So that's the end of my talk. I just wanted to thank a few people who made this research possible. My advisor, Dr. George Vandermerve at the University of Guelph, all of my lab members, Escarpment Labs who provided these strains and guidance on my project, Royal City who provided me with wort for this, and Diane and Armin at the mass spec facility who helped with the analysis. All right, thank you.
There we are. Okay, I'm back. Thanks, Caroline. I thought that was a great presentation. Hopefully people understand now that all the interesting things that are going on under the hood with Brettanomyces, and understand that it's a tough thing to study because every Brett strain is really different from every other one. I think Nate and I kind of got into this the tail end of the last presentation. We sort of talked about how every Brett kind of has its own personality, and you really can't apply the same rules from one strain to another. And in some ways that's frustrating, but in other ways that's also pretty exciting because really you can as a brewer you can develop a personal relationship with with one or two Bretts and really get to know them very well. And for those of us who are producing these cultures, it's really tough. I would say that's one of the more complicated jobs at Escarpment is growing Brett because you sort of have to get inside their head and recognize it's like, oh yeah, no, this one takes 3 days, but this one takes 2 days, but this one takes 3 days, but you have to add thiamine and fructose and there's all these fun little quirks with each strain that we've had to sort of figure out. And I think that's fun, but it's also it's a huge challenge, studying any of this stuff and getting consistent results. I think you put forth a ton of effort there, and I know that there's a lot of troubleshooting to even get to these results at the end of the day.
Yeah, quite a bit, but it was a fun journey and cool to see how it all came together in the end there.
Absolutely. Just want to mention we have one more day of our ScarpCon, little digital conference. Coming up tomorrow, we're going to be talking about quality control. So we're going to be talking about live cell counting demo with Chris Saunders. We're going to have tests you can do without a lab with Alex Mitro, and we're also going to be talking about diastatic yeast as well. So I know we had at least one question about comparing diastaticus to Brett here, and we're going to have more answers, more information about diastaticus tomorrow as well. So thanks again, Caroline. I do want to cover some of the questions we have. We have a few now, and people are now voting on the questions. If you haven't had a chance to vote on the questions, please do. This really helps us to sort of focus on what we need to answer, what you guys are interested in. So the first question, Caroline, if hopefully we're able to answer these ones. The first question is, and sometimes we can't, and a lot of the times we're sort of just pushing at the edge of the science here, so to speak. But the first question is, and this is a good question, does the alcohol that's produced by the Saccharomyces primary fermentation play a role in the secondary fermentation of each Brett?
In terms of whether it can ferment, or
maybe in terms of fermentation, in terms of flavor. Like, I thought it was interesting, for example, that when you did the sequential pitching, the secondary fermentations, and you saw that the Bretts that were pitched after Cali seemed to fare worse than if they were pitched after other yeasts. And I'm just wondering if there's something related to the sac that might explain that.
I mean, there definitely could be. We see some of the sacs can sort of ferment a bit better than others. Some of them leave behind more residual sugars than others also, which can, determine how well the Bretts can then ferment after the fact, and can lead to those differences we saw. It was extremely interesting to see, and not really what we were expecting, where you did really see the clustering of sacs. And then the other thing is there could be liberation of some additional sugars, whatnot, through flavor compound production, which could lead to some more sugars being present based on some of those yeast.
For sure, yeah. And it does, there did seem to be some trends associated with those combinations, for sure. So I'm not sure if the question is about the actual metabolism or the flavor production. I will say that just circling back on that interesting difference between Cali and the other yeasts in that sequential fermentation, I think there's an answer if anyone goes back to our yeast strain diversity talk. If you take that talk and this one and put them together, there's an answer in there. And if someone wants to come up with a potential explanation for why the Brett would ferment slower when Cali's around, shoot me an email and maybe we'll make a game out of it, or try to arrange a prize. I've got some ideas. Our second question is, I wonder how Brett uses wort fan for growth. Is it similar to sac? So less wort fan could bring more esters due to stress. Cheers from Brazil.
That's very interesting. So unfortunately, I didn't really get to look at the wort fan when I did these experiments. So I'm not entirely sure how what the contents were. It can make a difference in how these yeast are able to ferment, whether. I'm not sure how much fan exactly Brett uses in comparison to sac. Again, those were not experiments I ever really did. And then it was in terms of flavor compound production.
Yeah. Like would less fan impact the flavor? It could, and maybe some of those amino acid experiments that you did might help guide us toward that answer.
Yes. Yeah, I unfortunately don't have the volatiles for that experiment. We were just, that one was the last one I did before I wrote up my thesis, so it was just solely focused on that fermentation. But it could definitely have an impact there. I'm just not entirely sure what the fan contents were. We weren't measuring that, and it wasn't an experiment I built in, or comparing the two with that.
Yeah, I'm not sure how well that's been studied. Actually, that seems to me like a bit of a gap. Actually, nutrient consumption with Brett.
Yeah, there isn't too much on it. Again, I wrote my thesis 2 years ago, so the research definitely could have changed. I might not be as up to date as I was at that time, but there definitely wasn't as much on, sort of what kind of nutrients Brett requires or its nutrient requirements at that time in the research. And yeah, it would have been a good thing to build in, or an experiment, a follow-up experiment. It's just not something I had done at the time.
Yep. Yeah, and that's something that I think I can add just on the industrial side. This is total voodoo magic, but we propagate our Brett in fresh wort because it grows better in fresh wort versus reconstituted wort, like from liquid malt extract or dry malt extract. So we're not sure why that matters. There's maybe some difference in the micronutrients between those two things, but we've found in our industrial propagations of Brett that it really needs fresh wort, which really complicates our supply chain.
Oh, yeah, that's interesting. We always only ever used fresh wort for our experiments, so yeah. I don't have.
If you put it in a beer, like obviously it's built to survive. It's kind of like this microbial vulture, but yeah, to get it to propagate, in density, it's pretty needy.
Yes. Yeah. Definitely something we need to study a bit more. If these experiments showed us anything, there can definitely be things that help, but it's sort of still a knowledge gap.
Yeah. Here's another question. What explains the final gravity of Brett beers? Lambic finishes around 1010, 1.010. Some beers are bottle conditioned with Brett and don't finish at 1.000, but most American wild ales finish at 1.000. What might be the differences there?
So it could be in terms of, starting sugar concentrations, what sort of sugars are present in that wort at the start. We do see sort of different gravities even amongst the same kind of wort, like starting gravities for ours. How well it ferments out when it's taken out of fermentation, whether it's allowed to complete the fermentation or whether, it's sort of filtered out earlier than that, can all lead to differences there.
Yeah. Yeah, just to add sort of from our side, and this will come up tomorrow, I think, in the diastaticus talk, is that Brett seems to consume the starches and dextrins in slightly different ways, and actually maybe less effectively than some diastatic Saccharomyces strains. So I would guess that in some of those lambics there might be some residual dextrins or starches that the yeast actually can't ferment, and if those beers don't have diastaticus at any point in the process, then there actually might be some carbohydrates left behind that are not fermentable even by the Brett.
Yeah, it's also definitely a possibility.
What would be the best way to encourage fruity esters over phenolic aromas in a 100% Brett or mixed fermentation beer?
Oh, that's quite the question. Definitely warmer temperatures lead to, higher ester production. That's been well established. The things that we were seeing, I didn't show this, but a previous experiment was that the phenolic compound production seems to be pretty stable regardless of what you do, at least in our lab. Stayed pretty consistent, whether we had a colder temperature or warmer temperature. So really it's sort of just about bumping up that ester production, at least for us. Common way to do that is a warmer fermentation. So we don't, like temperature didn't seem to really affect how much of the phenolic compounds was produced for us.
Mm-hmm. It seemed like warmer temperatures, you got a little bit more.
Yeah. Yeah, so that wasn't part of the research shown here, but prior to ferment, we did do sort of a colder fermentation at 15 degrees and then one at 22. And you do see higher ester production there, but the phenols kind of stay the same at both temperatures.
Yeah.
Yeah, so that's sort of an easy way to do it.
How did you choose the amino acids that were supplemented in the supplementation experiments?
So there was a paper that came out, I don't remember what it was called, but it was a few years ago, a paper that came out that sort of tested all the different amino acids and the effect that they had on Brettanomyces growth. But this was in an aerobic environment. They also did an anaerobic, but they were solely looking at sort of how well it grew and whether there was a benefit in terms of the growth rate. And these three amino acids were the ones that seemed to have the greatest effect on Brettanomyces growth in anaerobic environments. So we were curious whether that sort of translated to a better fermentation rate, because they didn't study that in that paper. So they were chosen based on that prior research.
Yeah, I thought that was really cool, and that ties into a previous talk where in explaining yeast diacetyl formation, Nate was talking about the preferred list of amino acids for Saccharomyces, and that paper that you're talking about essentially showed that Brett has a different list of preferred amino acids. So even down to what kind of food it likes eating, it's different, right? So people like to compare Saccharomyces to dogs and Brett to cats, and this is correct in a surprising number of ways.
Yeah, it definitely likes to be unique, that's for sure.
Yeah, exactly. And yeah, just like cats, it's really hard to tell it what to do. And I think that's going to be our last question here, which sort of ties into that topic. And the question is, have you tested reproducibility of the same strain in different assays? Is each strain stable, even if the same batch is repeated? Thinking as a brewer, and also, what was the longest experiment you ran with the Bretts?
So to start, yes, we did do redoing these experiments to get sort of more reps in primary, secondary co-pitch. Especially that one was redone a few times. It's difficult in a lab setting just because we're sort of reliant on, for example, the wort we get and things like that, which, even if it's the same wort, it sometimes has a slightly different specific gravity starting, which can, make it a little bit difficult to have a direct comparison between experiments. And we've seen that, sometimes the incubator we're working with just for whatever reason might not be working quite as well during one experiment as another. So there's a lot of variables that come into play. So these have been redone. It's more so the way we look at them is to look at the trends between the different experiments, which do seem to hold up. More so than, we can say in a primary fermentation, Brett will produce this much ethanol and will produce this much of this flavor compound. That's really hard to say just because there's so many variables at play there in terms of, how much sugar we have to start with and things like that, which can change from batch to batch. It makes it sort of difficult to work with. And then what was the second part of that question?
So the second part of that question was, what was the longest experiment that you tried with the Bretts?
In terms of how long the fermentation lasted?
I think so.
So the longest I ever did was a 28-day fermentation. That's sort of, at the time I did a Brett primary, it was 28 days, unless it was in conjunction with like a SAC primary experiment. So like this primary, secondary co-pitch experiments were done for 21 days. Anytime there was a co-pitch component, it was usually 21 days. We wanted to find kind of that spot where we gave time for Brett to ferment, but also didn't leave the sac sitting for, way after it was done fermenting, which we still kind of had to. The longest I ever did was the 28-day fermentations for Brett primaries, though.
Yeah, I will say, yeah, I mean, we've gone a little longer on these things in the lab if we're doing like sensory scale trials. If we're, bottle kit, like one common thing we'll do is make a batch of beer and split it like in the bottles, because then we can just pipe that in like half a mil of Brett slurry, and you can split one batch through 6 or 7 different strains and really, get to taste what the different Brett strains taste like as a sort of a secondary addition in the bottle. And we've done those for, up to a year. And then, measured some of the differences between those just on the sensory level. And I believe we actually have a blog post coming out soon about some of those sensory trials. Some of Jonah from our team's experiments with digging through just stuff that he had isolated from different bottles of lambic and American sour beers, and seeing what's tasty and what's not, and that's always interesting. It's that there's so much diversity in Brett. There was one that tasted like ginger in one of his experiments, and you get all sorts of really cool stuff. We've pulled these Bretts out of the 40, 30-year-old bottles of Berliner Weiss, and some of those taste like white wine and citrus. Like there's all sorts of stuff going on, which is really exciting.
Yeah, definitely interesting. It's very good to do those kind of lengthier experiments and see how that goes. We're just a little limited with space, and we also just the volumes we use don't allow us to necessarily keep them for too long.
Mm-hmm. How do you feel about researching Kvike compared to Brett?
Way less frustrating in terms of how well Kvike grows really well in comparison to Brett, but it comes with its own set of challenges. Sometimes it's a little too fast. You miss things. They're hard to compare. I mean, Brett was kind of what started me in this world and in this research, so it's always going to have like a soft spot in my heart for it because of that. Definitely some interesting stuff we're working on now. It's nice to be able to wrap up experiments a lot quicker, not have to sort of wait a month to get results on things. But they're both very interesting and fun to work with for different reasons. Yeah,
absolutely. Just two interesting tools that we're trying to make easier to use for brewers.
Mm-hmm. Yeah, for sure.
Well, cool. I think that's it for questions. I don't know if anything else has popped up here. I think we're good for that. That's great. I just want to thank you again, Caroline, for coming and talking about your research. It's really fascinating stuff. Really glad that this stuff happened and that we're able to still collaborate on all this beer yeast research here in Guelph, Ontario. We're all just collectively trying to put Guelph on the map, and I think we're slowly succeeding. And one day we'll get there. Thanks again for coming and talking about Brett. For everyone who's in the audience. Oh, sorry.
Thanks for the opportunity. It was a lot of fun.
Yep. And for anyone who's tuning in, please try to come in tomorrow. Check out some of the talks we have about quality control. After this week, we're going to be likely taking a break for a week, and then we'll be back in here with a slightly new format. We're going to be aiming for one webinar on Tuesdays, and then we're going to do a brewer interview hopefully every Thursday as well, because we thought that our brewer interview with Barncat last week about hazy IPAs went really well, and that's a little bit easier than us preparing an hour-long slideshow. So looking forward to that. Looking forward to a little break, and then getting back and doing as much as we can to keep you guys learning while there's opportunity to learn and get back and get brewing. Awesome to see all these people from all over the world tuning in. I see lots of people from South America, Europe, US, Canada. Really cool to bring everyone together, and hopefully we can turn this into a little community and keep nerding out about yeast and science. So thanks everyone, and I'll see you tomorrow morning.
Yeah, thank you. See ya.